Hi, everybody. Welcome to our 13th lecture. Today, we will begin discussing glycolysis. If you like enzyme mechanism, I think you're going to have fun. Today, we will be uncovering the mechanisms of the first half of the glycolytic pathway. And as I walk you through the chemistry in each step, I want you to pay attention to the chemical logic of every transformation. Why does each reaction ultimately serve the goal of making glycolysis end products? And of course, why are they important? Our molecule of the moment is hexokinase, and this enzyme happens to catalyze the first transformation in the glycolytic pathway. Now, hexokinase is known as a housekeeping protein, and what that means basically is that its function is evergreen to the cell. It will constantly transcribe hexokinase because we constantly need to phosphorylate glucoses. And specifically, hexokinase will phosphorylate the hydroxyl on the sixth carbon. And in this schematic, we visualize what that transformation looks like. Hexokinase will take an equivalent of ATP and it will transfer a phosphate group from that ATP donor to this hydroxyl highlighted in red, yielding us glucose 6-phosphates, also known as G6P. And one of the reasons why hexokinase is a housekeeping protein is that G6P is locked into the cell. It cannot leave, which is good. Therefore, many metabolic pathways, not just glycolysis, begins with this enzyme. The KEQ equilibrium constant of hexokinase is 7.8 times 10 to the 2. Typical cellular concentrations of its substrates and products are 5 millimolar for ATP, 0.5 millimolar for ADP, 2 millimolar for glucose, and 1 millimolar for G6P. My questions to you are these. Is this reaction occurring at equilibrium? And if not, which side of the equation is favored, products or reactants? And most importantly, how does this benefit the cell? You first learned about glycolysis when you took general biology. However, I think it's going to be beneficial to you if I recap the basics before diving in any deeper. Glycolysis literally means sugar splitting. The sugar we are splitting here for energy is glucose. Fun fact, the old name for glucose used to be glycose, hence glycogen and glycolysis. But anyway, glycolysis is something that pretty much every organism does to make energy, which means this is an effective and ancient metabolic process that is conserved in all domains of life, at least to our knowledge. Even though glycolysis is considered the first step in cellular respiration, it occurs in the cytosol, not the mitochondria. We'll get to the mitochondria soon enough. The whole process of glycolysis occurs in a series of 10 enzyme catalyzed steps, and you can see that illustrated in this schematic over here. We start with glucose, and then 10 enzymes later, we end up with 2 3-carbon sugar acids called pyruvic acid or pyruvate. In doing so, in this conversion of the 6-carbon glucose to 2 3-carbon pyruvates, we will be generating 2 equivalents of ATP, cellular energy, for immediate use. Not a lot, but not nothing. And that's not all we're doing. We're cutting glucose in half, but we're also oxidizing it with NAD plus to form two equivalents of NADH. And we can carry off the NADH to use elsewhere in the cell. As for that pyruvate itself, we can also further process this for a lot more energy. Now, take note here that the entire glycolytic process is oxidation without oxygen. Therefore, we are not fully oxidizing glucose, we are partially oxidizing it. So it's a good start, but I would consider it to be overall inefficient. Doesn't make a lot of ATP or NADH. In a persistent lack of oxygen, this pyruvate molecule can be further oxidized anaerobically, and we call that fermentation. And it will become either lactate or ethanol and give you a little bit more energy, but really not a lot. Or if we were to have oxygen in aerobic conditions, we could fully dismantle, fully oxidize 1 glucose into 6 carbon dioxides, and this way produces the most energy. We will talk about these subsequent pathways in detail later. Before we get to any mechanisms, I want to skim over glycolysis's 10 steps. Then we will dive into each enzyme in greater detail. Personally, I like to think of glycolysis of having two phases. The first 5 reactions I like to call the investment or the preparative phase. And in the preparative phase, we will accomplish the following. We will consume 2 ATP, not gain. The delta G of the metabolic intermediates will go up, which means we are destabilizing the molecule or activating it. We will be committing the glucose and ensuing metabolites to the cell, and then the pathway itself. And we conclude our investments or preparative phase with making 2 glyceraldehyde 3 phosphates, or G3P for short. How do we get from glucose to this glyceraldehyde 3 phosphates? Well, let's take a look at this diagram. And I really encourage you, it's a lot of information. Try not to panic, pause and rewind as you deem fit. I think that'll be very helpful in internalizing all of the minutia. But we will begin with regular old glucose, and we will be adding in hexokinase, which will conduct our first priming reaction by phosphorylating it. This is where the first of our 2 ATPs will be spent. You gotta spend energy to get energy. And in doing so, the product of this is G6P, glucose 6 phosphates. And you can see on carbon 6, that hydroxyl now has a phosphate attached. So we've activated our glucose a little bit, and we've committed it to the cell. The next enzyme, phosphohexose isomerase, sometimes called phosphoglucoisomerase, it will do exactly what the name suggests, isomerize the phosphohexose. We will proceed from a glucose 6-phosphate to a fructose 6-phosphate. Fructose 6-phosphate. The third step is the second priming event, where we will take another equivalent of ATP and activate the hydroxyl on the 1 carbon as well, giving us fructose 1,6-bisphosphate. This has the added benefit of making the molecule more or less symmetric. We will have a phosphate on both quote-unquote halves. That's important because the fourth enzyme, aldolase, is going to cut that sugar in half into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate to rather high energy phosphotrioses. After we do this cleavage step, we will employ the fifth and final enzyme of the preparative phase, triose phosphate isomerase, which will scramble the dihydroxyacetone phosphate into G3P, which will converge these two molecules onto a single track, hence ending us with two equivalents of glyceraldehyde 3-phosphate, G3P, based off of a single glucose. The last 5 steps of glycolysis constitutes the payoff phase. Remember, since one glucose is cut into ultimately 2 G3P molecules, these last 5 steps occurs twice per glucose. Keeping that in mind, this is where we start to get the useful things out of glycolysis, such as 4 equivalents of ATP generated via substrate level phosphorylation, which is the manual transfer of phosphates from high energy molecules to ADP to make ATP. Additionally, we will be making 2 equivalents of NADH. The delta G of the intermediates will be trending down. The products will become more stable as we proceed through the pathway. And that pathway will end with 2 pyruvates. Let's talk you through these steps in a little bit more detail now. We will begin again with 2 equivalents of G3P, glyceraldehyde 3-phosphate, a very high energy phosphotriose. We will then hit it with glyceraldehyde 3-phosphate dehydrogenase. A dehydrogenase enzyme removes hydride. It extracts energy from the molecule, okay? This will constitute our first oxidation event and will net us 2 NADHs, one for every glyceraldehyde 3-phosphate, And because this molecule is so reactive, we're able to sneak in two inorganic phosphates for free, bringing us from G3P to 1,3-bisphosphoglycerate. There are now two phosphates on both 3-carbon sugar acids, hence the 2. The next step is catalyzed by phosphoglycerate kinase, and this is an example of our first ATP forming reaction. We phosphorylated and oxidized, previous step. Now we're going to cash in one of those phosphates on each bisphosphoglycerate, giving us 2 ATP. We spent 2 ATPs in the preparative phase, so at this point we break even energetically. Now we're left with 3 phosphoglycerate. We went from 2 phosphates to 1. The 8th enzyme, phosphoglycerate mutase, will relocate that phosphate to a more convenient position. We'll explain why later. And it moves it from the 3-carbon to the 2-carbon, shown over here. The 9th step, we will use enolase. And that is going to convert 2-phosphoglycerate into a very high energy, very unstable phosphoenol pyruvate. This molecule is so unstable that it's going to facilitate the final transformation, where pyruvate kinase will perform our second and final ATP forming reaction, and we will rip off that phosphate and end up with an incredibly stable, by comparison, pyruvate molecule, and we'll have 2 of them for every glucose. Now, taking a step back and looking at the forest for the trees, we released a lot of energy to the surroundings through this process. The total delta G of this pathway is negative 85 kilojoules per mole, which is quite a bit. However, remember when I said that glycolysis is effective for biology's purposes, but overall inefficient? That 85 kilojoules per mole extracted out is only 5% of the potential energy released from a glucose molecule. In the weeks to come, I will teach you how to get the other 95% out via aerobic respiration. And so begins the parade of enzyme mechanisms. We will be doing the mechanism of all 10 enzymes in the glycolytic pathway, starting with step 1, hexokinase. I will repeat to you all that this enzyme, what it does is it takes glucose, it takes ATP, and it facilitates a phosphoryl transfer onto the red shaded 6C hydroxyl position, converting glucose into G6P, glucose 6-phosphate, the phosphate shown explicitly over there. This reaction consumes ATP. You have to spend ATP to make ATP. And remember, what I'm very interested in you internalizing is the chemical logic of these glycolytic transformations. And the chemical logic of the hexokinase reaction is actually threefold. The first thing that it achieves chemically is it traps glucose in the cell. The cell membrane contains special glucose channels or glucose pumps where glucose can go in and glucose can come out. Just like so. When hexokinase phosphorylates glucose into G6P, it obtains a negative charge that makes it incompatible with that glucose transporter. So hexokinase commits the glucose to the cell. Related to that, hexokinase is effectively decreasing the concentration of free glucose. And I want you to think about this in terms of Le Chatelier's principle. Let's consider the glucose transportation process to be one that's in equilibrium, a dynamic equilibrium, with the concentration of glucose constantly being depleted. What we are doing is putting a stress on the system that favors increased importation of more glucose to sort of fill that gap. This will ensure that the cell is going to be constantly fed by external glucose. The third bout of chemical logic is that we are destabilizing the glucose molecule or increasing its delta G by adding in that phosphate. It's going to make it more reactive and it will facilitate future transformations in the glycolytic process. And not only that, we're going to eventually extract out again that phosphate in the form of an ATP molecule. Additionally, you should be aware that hexokinase is the first regulatory step of glycolysis. This means a couple of different things. It is very thermodynamically favorable and thus considered irreversible. Its delta G is very negative at negative 17 kilojoules per mole. And it sort of sets the pace of all possible reactions done on G6P activated glucose in the cell. We toggle the pace of glucose trapping or glucose activation through a process called product inhibition. G6P is the product, but it also inhibits this enzyme by binding an allosteric site, which will occur if the cell accumulates and does not use this activated G6P. And that just makes a lot of sense because if the cell is making G6P and not using it, well, there's really no need to commit it to that particular cell. Also of note with hexokinase, it's a really important enzyme, is these drastic motions that we are visualizing in this animation over here. The hexokinase enzyme is considered to be an exemplar of the induced fit model of catalysis. All hexokinases feature these two lobes that clamp down onto glucose and ATP. When the lobes clamp down, this will strip the substrates of their protective hydration shells by immersing them in a hydrophobic cleft. So we are stripping the solvation shells. And what's more, we're not only doing that, but we are burying all hydroxyls except for C6OH. This is how we get regio specificity. This is actually a huge deal. This hydroxyl and this hydroxyl and this and this and this are not dramatically chemically different. If we tried to do this in the laboratory, it's probably going to be more or less a crapshoot which hydroxyl gets phosphorylated. The enzyme, that's the beauty of enzymes. They can be precise, surgical precision. We're going to cover up every other nucleophilic hydroxyl except for the one we want to react. It's just beautiful. It is, in fact, considered to be a hallmark of kinases. Truly worth studying. Okay, it is mechanism time. The hexokinase mechanism is essentially a nucleophilic substitution. Our nucleophile will be the C6 hydroxyl on our glucose unit. And what will that nucleophile be attacking? An electrophile, specifically the electrophilic phosphorus atom on the gamma phosphate position of ATP. So what does that attack look like? I will start by drawing out our ATP unit. I will symbolize the non-reacting adenosine base as ADE in a box. And then I will draw out the triphosphate moiety explicitly. I will add in several oxygens. And these oxygens on the gamma and beta position will chelate Mg2+, and that chelation will activate the phosphoryl anhydride for nucleophilic attack. So what does this attack look like? You may recall from the serine protease model that the hydroxyl group is not a terrific nucleophile on its own. We need the enzyme to jack up its nucleophilicity. And a tried and true strategy for doing that is acid-base catalysis. We can achieve this through a general base, which is any amino acid that can behave in a base-like manner. It's often signified as just the letter B with a lone pair. And that B will attack the H on that hydroxyl, regio specifically. It will deprotonate it and form an alkoxide. Alkoxides are very reactive, very nucleophilic. We can now attack the electrophile, specifically this phosphorus. When we do so, we are establishing a new bond between the C6 hydroxyl and that central phosphorus. We have to now break a bond. The bond broken will be this pi system over there. This may sound a little bit familiar. Those electrons will rise up as a transient negative charge, and then it's going to collapse back down again. And once it does, something needs to break. It could be the bond that we just formed between that phosphate and that glucose. If it did, we just go back a step and try again. Not the end of the world. In theory, you could break one of these PO- bonds, but I think that's improbable because O- is not a great leaving group. You want to know what is a good leaving group? ADP, specifically the pyrophosphoryl moiety shown over here. If we break that apart, we will get a lot of resonance, which is good, very thermodynamically favorable, irreversible, that will drive this reaction forward. This is the bonds that we break. And what happens when we break that bonds? We transfer that gamma phosphate right over there on G6P, and what gets left behind is ADP. With Mg2+, of course. The next step is catalyzed by phosphoglucoisomerase. This enzyme takes our freshly made G6P and converts it to F6P, fructose 6-phosphates. The goal of this enzyme is to take our glucose, which is an aldose, and convert it to a ketose. And the reason why we are doing that, the chemical logic, is that it will activate the third carbon for a cleavage step that occurs later on. And this happens because we are moving C3 over here, which is right next to the hemiketal, which is pretty reactive. This will make more sense later on, why that's activating. The reaction delta G, if you've noticed, is slightly positive here. It is positive 1.7 kilojoules per mole. So it's a little bit unfavorable. However, the third step of glycolysis coming up is so favorable that it will help drive equilibrium towards the products of this reaction. Said another way, step 3 is going to use F6P as a reactant so quickly, it doesn't have time to revert back to G6P. Again, it's Le Chatelier's principle. The concentration of this is going down, which will stress equilibrium into the forward direction, and thus help us form products. Keep glycolysis going forward. Let us now unpack the mechanism for phosphoglucoisomerase. The very first step is to induce a ring opening reaction of the glucose reactants within the active site, thus linearizing it, like so. Once it is linearized, the next thing our enzyme will do is take a basic residue, B, and it will do an elimination. It will eliminate the proton highlighted in red on the 2C position. This will redirect the electrons between the carbon and hydrogen bond onto the C1 C2 position. This is going to form a cis-ene diol. The hydroxyls on carbon 1 and carbon 2 are cis to each other, and they're going to be attached to an alkene. Hence, it is a diol and alkene, cis-ene diol. Enols, you may remember from organic chemistry, are pretty unstable. cysteine-diols are way worse. They are short-lived functional groups, and that is going to facilitate the next reaction. The hydroxyl on the C2 position is going to undergo a rearrangement where a general base will prompt dissociation of that hydroxyl hydrogen, and the electrons from that bond will hop on down over there, which means we're adding a bond to the C2 position, which means we need to break one of the bonds associated with C2. How about we break the pi bond we just established in that cysteine-diol group? That pi system will break and act as a nucleophile and attack the H that we just abstracted from the C2 position in the previous step. Once all is said and done, what we are left with is this fructose unit, which is now linearized. It is now free to dissociate and recyclize into fructose 6-phosphate. I want to briefly pause and rewind here because I know this is quite jumbled looking. Let's go through this mechanism one more time, but let's just track the position of the double bond migrating throughout the molecule. And I will highlight this in a different color. Let's try blue. Originally, we are dealing with an alkane backbone, and our double bond is in a terminal position, hence we have an aldose. This elimination is going to move that double bond down over here, inch by inch. The second bout of acid-base catalysis will now migrate that double bond again to where that C2 hydroxyl used to be. We go from the aldehyde position to an alkene, and then resaturating the alkene brings us to a ketose, specifically fructose. It's quite elegant, in my opinion. Step 3 is catalyzed by phosphofructokinase, and it's perhaps the most important step in glycolysis. This is our second ATP-consuming reaction, and we will use this ATP molecule to phosphorylate another hydroxyl on our sugar unit. We will take F6P and now phosphorylate the hydroxyl on C1, yielding us fructose 1,6-bisphosphate. The chemical logic here is twofold. First and foremost, and shown in bright pink, it traps sugar to glycolysis. Hexokinase commits that molecule to the cell. Phosphofructokinase commits it to glycolysis. There's no going back after this. We are destined to become pyruvate whether we want to or not. Secondly, this reaction will provide symmetry to our metabolite, which facilitates parallel metabolism of two cleavage products down this pathway. Very soon, we will be cutting our molecule into two trioses right over here. And by adding a phosphate onto the one position, it ensures that both halves will have an activating phosphate on it. what makes this step such a powerful gatekeeper, such a powerful choke point, is its, again, very exergonic nature. It's very negative, very low reaction delta G, which is around negative 14 kilojoules per mole. And since phosphofructokinase gatekeeps glycolysis, nature has installed many allosteric regulatory sites on this enzyme. Our upregulators are going to be ADP, AMP, and another molecule that we'll discuss like 1 or 2 lectures from now called fructose 2,6 bisphosphates. This is a signal molecule that the body will create when there is a high energy demand. It also stands to reason that ADP and AMP, which are depleted chemical batteries, would also stimulate the activity of phosphofructokinase, because that's a sign that the cell is running out of energy. We need more. We need more glycolysis. These are the activators. Now, what are PFK's inhibitors? Stands to reason that ATP, which is, make no mistake, a substrate of this enzyme, it's also an allosteric inhibitor. When ATP concentrations get very high, that means the cell is not using the energy it creates. So there's really no need to burn that sugar. This process destroys glucose, and we can use that for other purposes. So let's tamp down the glucose furnace, so to speak. Another allosteric inhibitor of phosphofructokinase is going to be citric acid or citrates. And this will become more obvious when we get into lipid metabolism and even the citric acid cycle. But this is a metabolite that will accumulate also in anabolic conditions where we have a surplus of metabolites and resources, and thus there's less of an impetus to break molecules down for energy. There's quite a bit of regulation occurring at this step. And so I think, again, this reinforces the fact that phosphofructokinase is not only what commits glucose to oxidation, but it's the pacemaker of glycolysis itself, or at least the most significant one. Now, as for its mechanism, you will be quite pleased to learn that it very closely resembles that of hexokinase, which you are already experts in. So we can just proceed. Step 4, which is catalyzed by aldolase, is a very dramatic reaction. This enzyme will take our fructose 1,6 bisphosphate, which is properly committed to glycolysis now, and it's going to split it in half into 2 very high energy phosphotrioses. That is the point here. We are going to be greatly destabilizing these metabolites moving forward, which are specifically dihydroxyacetone phosphate, DHAP for short, and G3P, glyceraldehyde 3-phosphate. One's an aldotriose, one's a ketotriose. The fact that we are destabilizing and activating these compounds is inscribed in the fact that our reaction delta G is super positive. Positive 24 kilojoules per mole. Thermodynamically, that is incredibly uphill. And therefore, it's going to be considered reversible. or hard to achieve. However, though, the delta G of this reaction in the cell, not in a test tube, but in real life living organisms, is closer to 0, which is a little bit more doable, a lot more doable. And that's because, again, the next step is so fast that the products don't accumulate long enough to go into the reverse direction. Specifically, the next reaction is going to eat away at DHAP and convert it into G3P. And that's a very common theme here in these metabolic pathways. Reaction delta Gs, when very positive, the next step is usually fast to compensate. And pull reaction equilibrium forward. The aldolase mechanism is the most complex in my opinion, but if it helps, what we're essentially doing is the reverse of an aldol condensation from organic chemistry too. The first thing we will be doing to fructose 1,6-bisphosphate is again to linearize that molecule within the hydrophobic clef. And we can see that right over here. We go from a cyclic structure to a linear structure. And once that linearized sugar is buried in, we're going to get to work. The first attack will be conducted by a reactive site lysine, shown over here. Lysine is nucleophilic, and the carbon on the nearby carbonyl is going to be electrophilic. This C2 carbonyl is the one that we made, by the way, 2 enzymes ago in the isomerase step. Isn't that convenient? The lysine will attack that carbon and form a bond with it. And since we're adding a bond to that carbon, we need to break one of the other pre-existing bonds on it. And the one that's going to go is the pi bond in that carbonyl. It is going to migrate over to a hydrogen on a general acid residue on the other side of the binding pocket and converts the ketone into a secondary alcohol. And with that lysine nitrogen bound to that carbon, this will give us a tetrahedral intermediate as a consequence of step 1, shown over here. What happens next? This tetrahedral intermediate will undergo an additional rearrangement, this time prompted by a general base vicinal to our reactive site lysine. The general base will attack one of the protons on our bound lysine, and that will cause the electrons in its NH bonds to move over onto our NC bond. This is going to form what's called a Schiff base. Which is the same thing as an imine. Not an amine, an imine. We are again increasing the bond order on that C2 carbon, which means another bond has to go. The other bonds that we will be destroying is also one between the carbon and the oxygen on the hydroxyl we just made. These electrons will seek out a proton via acid-base catalysis, and it will completely remove that OH and dehydrate it as a water molecule that will diffuse away. And this finalizes the formation of our Schiff base. The Schiff base is a very convenient handle for enzymatic covalent catalysis. For one reason, we have just formed a covalent but reversible bond with our substrate, which helps reel it in and reduce entropics. It will also prime us for the next step by serving as an electron sink. I'm going to loop back to that very soon. Remember when I said that converting glucose to fructose activates the third carbon, which is right over here? This is how. That third carbon is between three highly polarizing groups. It has a hydroxyl group on C3. On C4, there is also a hydroxyl group. And on C2, it is next to a Schiff base. So it's right smack dab in the middle of three pretty exciting entities. What is the consequence of that? Well, a general base on the other side of the active site, over here, will attack the hydrogen of the hydroxyl on the C4 position. This, it's like dominoes, will cause the electrons to fall down from the OH bond to the CO bond, turning it into a carbonyl. Oh look, we increased the bond order on our already tetravalent C4 carbon. That means we have to break a bond to make sure we're satisfying the octet rule still. Which bond are we going to break? Well, normally it's really hard to break carbon-carbon bonds. But remember what I said, C3, which is right next to C4, is in the middle of three very polarizing groups. The electrons from that bond will break apart and migrate to the bond between C2 and C3. Just like so. It's getting a little crowded in there, I know. Once we do that, remember how the Schiff base is acting like an electron sink? It's facilitating that motion. And it will, in turn, relieve some of that pressure by moving the pi system here between N and C, that double bond, and move it back onto that nitrogen, that positive nitrogen. This is a very confusing step, so I like to break it down into two forces. Over here, this is acting like an electron pusher. This Schiff base is acting like an electron puller. And what gets broken between the pushing and the pulling is the bond between C3 and C4 broken. This entire bottom portion, by the way, is G3P, glyceraldehyde 3-phosphate. It's not attached to anything, so it's free to leave. That is the first product that gets released through the aldolase mechanism. From this point on, it's all downhill from here. Our goal from this point forward is to undo the shift base, holding in the top half of the molecule into the active site still. The general base over here is going to now function as a general acid. The lone pairs on that nitrogen of the lysine, that lone pair will fall down and it will cause this double bond to saturate. And it will take that hydrogen from BH, bringing it back to just B lone pair. Once we have resaturated that double bond to an alkane, the last step is hydrolyzing the shift base. Water, which was released in step 2, will come back in, and it can destroy shift bases really easily, reverting it into a carbonyl. This one over here in our second product, DHAP, dihydroxyacetone phosphate, bringing us back to square one, an empty pocket with a deprotonated lysine and these 3 other residues. Take a deep breath. I know that was quite a bit. I encourage you to wind back and look at this again. The 5th step of glycolysis, catalyzed by triose phosphate isomerase, marks the end of our preparative phase. Yes, we have just reached the halfway point. And the halfway point is actually in a bit of a pickle. Because of the previous aldolase reaction, we have 2 different substrates to work with, and we need to oxidize them both with the same set of enzymes into pyruvate. This is going to be difficult, so what glycolysis is going to do is make our lives easier. The 2 products of aldolase, DHAP and G3P, they can be interconverted with triose phosphate isomerase. And more specifically, this enzyme is going to convert the DHAP into G3P, thus reconverging glycolysis back onto one track. So moving forward, G3P will be the sole substrate moving onward. This, however, has its own set of challenges because the 5th step reaction is a little bit unfavorable. It has a positive delta G of 7.5 kilojoules per mole, which isn't nothing. So equilibrium is actually going to favor the formation of DHAP, dihydroxyacetone phosphates. However, we have a trick up our sleeves. The next step is going to consume G3P so quickly that Le Chatelier's principle will kick in. G3P will be a short-lived intermediate, and thus we are going to stress equilibrium to proceed to the right. Therefore, yet again, while the experimental test tube delta G of this reaction is positive, the cellular delta G is closer to 0. The mechanism of this isomerase actually closely resembles that of phosphoglucoisomerase, which means we will form a cis-ene diol to move the carbonyl from the 2 position to the 1 position. You should, however, be broadly aware of how to map the original 6 carbons of glucose onto our DHAP and G3P because of how aldolase cuts fructose 1,6-bisphosphate down the middle, like so. Aldolase cuts right over there between carbons 3 and 4. So keeping track of this, I think, can be a little confusing. Let's begin examining this graphic over here, which I think outlines it really well. In the linearized version of fructose 1,6-bisphosphate, we have the top portion, which is destined to become DHAP, split between 3 and 4, and the bottom portion is destined to become G3P. So we're going to move the red and the blue portions down after we use aldolase to cut. And so carbon 1 of DHAP was also carbon 1 on F16BP. This ketone lines up with carbon 2. And then the hydroxyl lines up with carbon 3. Triose phosphate isomerase is going to move this there and the hydroxyl into the secondary position. Once we do that, we essentially have converted DHAP into an upside down G3P. And you can see that when we look at the blue box on the right. The 1 carbon on G3P was derived right over here from that 4 carbon on glucose. This is also going to be the same thing as carbon 3 from DHAP. Highlight that. And we're going to work backwards. Carbon 2 on G3P came from carbon 5 on glucose, and it also comes from carbon 2 on DHAP after triose phosphate isomerase again does its thing, leaving us with the 3 carbon of G3P, the one that's phosphorylated. This comes from the last 6th carbon on glucose and the 1st carbon on DHAP before we isomerize it. All right, this is tricky at first blush, but just try to imbue it in your brains that the equivalent carbons are ultimately 4 and 3, 5 and 2, and 1 and 6. Let's pause things for now by reiterating that step 5 is the halfway point to glycolysis. It is from here onward we are going to change our philosophy from jacking up this metabolite with high energy phosphates to getting a little bit of payoff. And the next 5 steps is where we will observe these substrate level phosphorylations that make ATP and these non-oxygen based oxidations that will generate us NADH. Pat yourselves on the back. This is one of the harder lectures, and I think you all did a great job. Until next time.